Next up, we have Oleander Medical Technologies. Get this going here. Developer of a cancer treatment technology designed to destroy cancer cells without damaging other cells in the body. The company's technology is a treatment for late-stage cancer that does not rely on surgery, chemotherapy, or radiation, and kills metastatic cancer cells by forcing them to take in sodium ions while blocking the cells from pumping the ions back out, enabling medical researchers to treat cancer naturally, painlessly, and cost-effectively, unlike radiation and chemotherapy treatments that, as we know, kill both normal and cancerous tissue, resulting in potentially serious damage to the individual being treated. Today we have Ms. Paige R. Miller, Esquire. Ms. Miller, a second-generation technology entrepreneur, has focused her career on leading enterprises to plan and deliver challenging, innovative projects. She served for 18 years as commissioner of the Port of Seattle, one of the nation's largest combined airport and seaport authorities, where she was a leader in the successful effort to add the third runway at Seattle-Tacoma Airport and to transform the Seattle waterfront. She also was the founding chair of Port Jobs, a nonprofit enterprise that has connected nearly 30,000 people to jobs in the port and aviation sectors, with special focus on groups traditionally underrepresented in those fields. She has served as a board member of a privately held real estate development company that brought the largest mixed-use residential development to downtown Seattle, and as executive director of the Arboretum Foundation, where she led the fundraising for major new exhibits and infrastructure projects. Previously, she was in private law practice in Seattle and in Philadelphia. Ms. Miller is a graduate of Brown University and of Yale Law School. She is a member of the Washington State Bar Association and serves on the board of Port Jobs. She brings the company expertise in fundraising, budgeting, critical path project management and delivery, governance, strategic partnerships, and external relations. Super excited to hear about this one.
So what am I doing working on cancer after all of that, right? This is really an exciting way to look at cancer and to treat cancer that's unlike anything you guys have seen before. The conventional wisdom for treating cancer, which we all know, runs something like this. The most effective treatments are narrowly targeted to a specific cancer subtype. They target at the genetic level, not at the cellular level. They destroy a specific component of a cancer cell. And of course, they are best administered at an early stage of the cancer. In addition to that, the best new therapies, such as immunotherapy, are drugs that are very expensive to develop, as we just heard, and take a long time to be approved. Could there be a better way? Introducing targeted osmotic lysis, or TOL. So this is a revolutionary, novel treatment for advanced cancer that combines a generic drug, digoxin, foxglove grows in my backyard in Seattle, and a proprietary pulsed electric field device. TOL has the potential to be an effective therapy comparable to a new drug, but with a much shorter path to regulatory approval. It has significantly prolonged life in dogs and mice without serious side effects and without damage to normal tissue. It was used in one case in 2020 in emergency use with no side effects, and we have just started in January our first pilot studies in human patients. So the slide you see on the right is of a mouse. Mouse was inoculated with triple-negative breast cancer. Tumor was allowed to grow, then treated with TOL, then sacrificed. So what you see is just under the skin, all the blasted tumor, and below that, beautiful, nice, undamaged, striated muscle. So the research path for this treatment started in the lab with tissue culture, lung, breast, colon, prostate, pancreatic cancer. all were lysed within less than a minute, 95 to 100%. The scientists then moved on to mouse studies, mostly with triple-negative breast cancer, and they found they could double survival time compared to controls using TOL. We then moved to making sure it was safe in animals with a safety study at LSU Vet School. That was 2019. Then for 4 years, 20 to 24 ongoing, we've done clinical studies in dogs and some cats at 2 local veterinary hospitals. We found we could reduce tumor size, prolong life, and not create harm. Last fall, we began a toxicology study with a CRO. We are about to get that report. And we've started our studies with humans. This year, we expect to be doing clinical trials by later in the year. If there's one slide for you to pay attention to in this deck, it's this one. These are the cancers we think are potentially responsive to TOL. Virtually all carcinomas, a very few sarcomas because they mostly don't overexpress sodium channels, and mesothelioma. So the ones in red have been tested and efficacy seen in either tissue culture or mice or both, so breast, lung, prostate, pancreas, colon, and mesothelioma. The ones in green, we've seen effectiveness in dogs, so adenocarcinoma, nasal, oral squamous cell, and bladder actually, but we're now doing that in humans. So how does this work? Cancer's Achilles heel is that it overexpresses sodium channels and pumps at late stage compared to normal tissue, and this can be used against them. So here we've got a cell, endocellular membrane, and sodium ions inside and outside the cell in resting concentrations. Here's a sodium ion channel, voltage gated, a little bit of stimulation, and in goes more sodium. So we now have an imbalance. The ATPase pump will kick out the excess sodium and restore the resting balance. So that's a normal cell. What's been known for a very long time is that advanced cancer cells, again, mostly carcinomas, way overproduce the number of sodium channels and pumps. This is showing 3 times, think 10 to 50 times, that membrane is going to be littered with sodium channels and pumps. So a little stimulation, way more sodium goes in the cell, and the pumps kick it out to restore the balance. So that's a viable cancer cell. Targeted osmotic lysis is ridiculously simple. You give the patient a drug to block the pumps, in this case, cardiac glycoside, digoxin, so the pumps are locked down. Then you put the patient in a device that will repeatedly pulse to open the channels. So you flood sodium into the cell. Cellular mechanisms don't work very well with that excess of sodium, so the cell has a workaround. Osmosis, that thing we all learned about in high school. So water comes across by osmosis to dilute the sodium concentration, and we keep pulsing, and we keep bringing in more sodium, more water. Of course, the cells expand with all that extra water, and they expand and they expand until they lyse. Really simple. Okay, you're all going to ask me, what happened to the normal cell while you were doing that to the cancer cell, right? Normal cell got the same drug, getting the same stimulation, but so many fewer ions enter, so much less water enters, that less swelling occurs, the membrane's stretchy. When the drug wears off, out goes the extra sodium, out goes the extra water. We do not damage normal tissue. I want to repeat that. We do not damage normal tissue. Okay, so this is a combination product. The drug, everybody knows. Digitalis has been used in medicine forever, or at least since 1785, when Dr. Withering first wrote about it. It was first approved, digoxin itself was first approved in 1954. It's well understood to be safe within its approved, which is narrow, therapeutic range. It's dramatically shorter for us to get regulatory approval because this is a new indication, it's not a new drug. And it's used in exactly the same dosage as if we were using it as a heart therapy. Now, the device is brand new. This is a coaxial ring, an electrostatic pulsed field device, fully licensed to our company. Both the prototypes you see here use 110 volt wall power. They don't generate any heat. They don't require any elaborate shielding. They don't require any elaborate installation. The four of us who are the principals in this company have all been responsible for setting it up multiple times. It's like a great big Lego. So I want to talk a little bit about our companion animal results and then humans. So again, we tested the safety at LSU Vet School before we then went to treating people's pets with advanced degree disease that were out of options at two animal hospitals here, Metairie Small Animal Hospital and Monroe Street in Mandeville. We've treated more than 40 animals with these and had no serious side effects. I want to tell you about just a few of them. So this is Dodge. Dodge was a 15-year-old neutered male with 2 masses in his right lung, diagnosed as bronchoalveolar adenocarcinoma, a rapidly progressing cancer. He had no prior cancer treatment and no other cancer treatments while we treated him. So we started in September 2020 and treated him for 7.5 months. What we found was his two tumors shrank about 10% and then did not grow for the entire 7.5 months. Now, he was an old dog and he had severe arthritis and dementia, and the decision was made to euthanize him for those things, not for his cancer. The vet did a necropsy one Saturday morning and called us all excited about 8 a.m. What he found was that the tumors were these gelatinous messes, but not solid hard tumors. Sent the tumors to the pathologist, 90% necrotic. One way we test for whether cancer's likely to respond is we do immunohistochemistry on a tissue sample. If it lights up bright green, lots of sodium channels. So Dodge is lit up bright green. After he was treated and euthanized, not much in the way of sodium channels. This is Marley. Marley's a black Labrador. She was diagnosed with a really nasty oral squamous cell in 2021, October. We started treating her in December of 2021. Tumor grew some, then it was debulked by the vet, and thereafter only TOL treatment. So you can see from April of 22 to June of 22, 2 months later, the tumor is dramatically smaller. And by that summer, there was no sign and pathology of the tumor. We kept treating her until July last year and then just stopped. The dog is still alive. She is doing great, so more than 2 years after treatment. Otto. Otto is a Rottweiler with bladder cancer. The vet thought he had 2 to 3 months because of the size of his tumor and the likelihood it would block his ureters. We've been treating him since July of 2022. He's never seen, the vet's never seen a dog last more than 5 or 6 months with a tumor that big. He's now at more than 19 months, and the slide is wrong. We are still treating him. So turning to the human market. We all know this. 1.7 million people are diagnosed with cancer each year in the U.S. More than 600,000 will die. I listed on the left cancers that we have successfully lysed with TOL: lung, colon and rectum, pancreatic, breast, prostate, mesothelioma. And then the number of people who die of each of them each year, and you'll notice it adds up to more than half of that 600,000 just with those cancers. So our first human patient was in November of 2020. She had a very advanced cervical tumor the size of a softball. She was in hospice. She probably had 3 to 10 days to survive. We did an emergency use treatment here at University Hospital. We got to do 2 days of that. She had dramatic results in that her appetite, energy, and color all improved dramatically. We know that the tumor had more necrosis, and she lived 9 weeks, about 8 more than was expected. We did not get an opportunity to do another cycle of treatment. It's the biggest regret I have in that long career that you heard about, that we were not able to get to treat her again. So we are now doing a pilot study offshore. We started January 10th at a clinic in a jurisdiction that would allow us to export the device parts there under an IRB. It's in a special economic zone, and it's run by 2 Americans. So I've listed here the kinds of cancer that have been treated so far. All of them are very late stage, with the exception of one. And I want to tell you about him. So the study is obviously designed to get safety and tolerability, but beyond that, we are hoping to help some patients who have no other options. They've run through all the standard care. And we also want to figure out which cancer of the many we might try we can use for our first in-human trial here in the U.S. FDA has said you can do a first 15 patients, assuming the tox study comes out okay, but only one cancer. So we're trying to figure out which should be first. All of the patients, again with one exception, have stage 4 carcinoma that's not responded to standard treatments, and they're enrolled in a protocol that we designed based on what we learned from the companion animal and laboratory mouse studies. The current design is to get a steady state of digoxin aboard, then to treat for 2 consecutive days, 2 hours each day, weekly for 3 weeks. We get images before and blood work before. We will then get images after. And because of inflammation, you've got to wait a couple weeks after treatment before you can do your post-treatment imaging. So we don't have a lot of that yet. We're also looking at endpoints for quality of life, imaging, and measures of tumor viability. So with quality of life, we're looking at appetite, energy levels, sleep. Actually, we found people's hair has started to grow back, which is sort of interesting. And then also imaging for tumor size, numbers, density, and comparison with anticipated pretreatment survival and actual survival. We've treated about 14 patients so far. 9 have completed the first cycle of 3 weeks of treatment. 2 withdrew. One of them went into hospice. Patients experienced tingling or pinprick sensation in proximity to known tumor sites, and some places they didn't know they had tumors. The sensations have diminished with subsequent treatments. Visible tumors have expressed serous fluid or have sloughed off, leaving minimal evidence of the pre-existing tumor. And palpable tumors or lymph nodes have noticeably decreased in size. Similar to the observations in the companion animals, patients consistently report increased energy levels, improved appetite, and improved sleep, not typical of somebody who's in cancer treatment, I would say. Preliminary imaging and laboratory results are consistent with increased evidence of tumor necrosis. So this patient I wanted to tell you about. He's one of the inventors. He's sitting up here in the second row. So Dr. Paul has squamous cell carcinoma on his face. He's had repeated surgeries to excise tumors, and he comes back all bandaged up. He took the opportunity while he was offshore with Hank Mills setting up the device to treat himself. He noted the tingling sensation at the tumor sites at about 15 minutes into the treatment, and that some tumors leaked serous fluid. He also noted that his lesions turned quite bright red at about 20 minutes into his first treatment. So that top picture is in the device the first day he was treated. He did a second treatment for 2 hours on the second day, and he has managed to get a couple of sets of treatments since he's come back. The picture below is 6 weeks after the first treatment. His tumors have sloughed off. They've left no scars. I told him that I would invite you to examine his face, and he said yes, he would let you do that. I mean, this is a place where it doesn't require a CT scan or an MRI to see what the difference is from this treatment. So this is the core team. Hank Mills and I, and Hank, raise your hand, are the business side. Harry Gould and Dennis Paul are the co-inventors and scientists. They're both on faculty at LSU Health Sciences Center. We have an expert in veterinary health to help us with our CRO study. Dr. Bruce Burlington got his MD degree here at LSU. He was a director of CDRH, the device center, a while back. He's been invaluable in helping us figure out how to navigate FDA. Of course, it takes more than 6 people to do this, so this is the slightly bigger team. We have more FDA experts, we have oncologists, we have attorneys. I'm not the attorney, I just know how to hire attorneys. Phantom Laboratory in upstate New York is our device designer and manufacturer. They've been terrific. Key Consulting has provided services for the offshore treatments, And again, we've worked with the 2 animal hospitals, particularly Metairie and Dr. Brian Melius. The treatment is patented in the U.S. and a bunch of other places. A second patent was issued in the U.S. last year and in the European Union last month. This time we also went to China. There's patents are pending for the 2 types of devices, and those are also licensed to us. Milestones. This has been a long process. The first that the guys had the idea this might work was in February 2011, so a long time ago. Because it is so outside the box of anything in the regular cancer world, there's been no federal funding from NCI, from DOD, from anybody. I mean, they're 2 pain doctors who think they've come up with a way to treat cancer. So all of the money has come from private investment, mostly individuals. A lot of them are doctors, actually. One foundation, the LSU Health Foundation, has invested in us. The first technology they invested in from the Health Sciences Center. So to recap, we've had the device bench tested at an electronics laboratory. It passes all the safety stuff. There are a couple of tweaks, like what our labeling is, that need to be fixed. The safety study was done last fall. The report should be coming within the month. We've started our overseas human pilot studies in the Caribbean. We expect to apply for an IDE this summer and to begin our first in-human investigation this year, and then to do trials for the next couple of years. Unlike the drug side, the path, the number of people you need to treat on the device side is a much smaller group. So we should be getting to market faster than if this were just a drug. So, unconventional wisdom for treating cancer. I've done a markup here. The most effective treatment might be a treatment that targets many solid tumor cancers, that targets at the cellular level, that uses a specific feature of a cancer cell, and is best administered at advanced stage when the cancers overproduce the sodium channels. The best new therapy may be a device that is cheaper and quicker to develop and to get regulatory approval. Indeed, there may be a better way. So we will be doing another round. We've raised all of $7.3 million to get this far, if you can imagine that. But we're about to do a round of something between 3 to 5 million later this spring. Hank Mills and I, this is our contact information if you're interested in learning more about targeted osmotic lysis. Thank you so much for listening, and I think we have a couple minutes for questions. You can't tell me there aren't any questions about this. Yes. So, most people don't have pigs or sheep as pets. But besides that, dogs are a good model when you're using digoxin. Their cardiovascular systems are very similar to humans. And actually, when we went to the FDA, there was question, why are you using beagles? Why aren't you using some bigger animal? Well, the problem is, there are pigs. Their body composition isn't close to human, way more fat. And besides, they wouldn't sit in a cage for 2 hours, so you'd probably have to sedate them, so then you've added something. There were sheep, but a whole bunch of sheep is stomachs, and wool, so they don't really fit the same body type. Turns out the dogs truly are the best model, and because of public outcry, the only dogs you can use for studies these days are beagles. Yeah, well, the other thing is we have talked about going into the vet market. The FDA does not regulate devices for animals that are not eaten by people. So, but because the human market is so much more lucrative in terms of having insurance and people being willing to pay, that's where we're starting. We probably will circle back to the vet market. Some high-end cities, probably people would afford to pay for their pets. All the ones we're doing now, we've done for free. Yeah. And the answer is yes, but digoxin is the only one already approved by the FDA. So yes, there are other cardiac glycosides. Dr. Paul is itching to tell you, but no, you can't, because we don't have any patent applications in to use them. Yeah, but that means going up the drug side and the long path to get there. And if there's anything I can tell you about me, and these guys will agree, I am really focused on getting us to market. Once we've got some revenue coming in, we can go back and look at all kinds of ways to make this better. But right now, it's about getting something that really works to market.